Kristen Stewart. Let me get to my presentation. Sorry. Okay. I decided to do the regulation of gastric phase of digestion. In my slideshow, I will go over each of these sections. It outlines the three major functions that are within the gastric phase of digestion and how each help the regulation of digestion. So the first is storage regulation, which is done by the stomach. As noted in Silverthorne, when ingestion is more than we need from a nutritional standpoint, the stomach must regulate at which food enters the small intestine. That makes sure that the stomach is not overwhelmed, or the small intestines are not overwhelmed. This mechanism is a very helpful tool for the small intestine. It allows the small intestine to do its job properly. That job is reabsorption, secretion, and digestion. So it is nice that the stomach does that. The way it does this is by relaxing and getting bigger, allowing more food to sit in there. Next is gastrin secretion. Gastrin secretion promotes the release of acid. It comes from G cells, which produce gastrin, which is a hormone and is released into the blood. It is stimulated / regulated by many factors. The first couple of factors is presence of amino acids, peptides, and by distension. That comes from the earlier on phase of digestion. And then there's neural reflexes in the short reflex of the ENS, which is gastrin releasing peptide. This peptide is a huge factor in connecting with the G cells to produce gastrin. I will go on to the next piece where I talk about acid secretion. This is huge for the stomach and digestion, and it is important that our body regulates this. And without it, we won't be able to digest. So it is an important piece for homeostasis. The acid comes from the parietal cells, which secrete HCl, which is gastric acid. This little picture in the bottom right hand corner is super helpful for me when learning where and how the components come into the lumen of the stomach. As you can see, the hydrogen is created from the H2O, which breaks it down, and the bicarbonate goes out and brings in the Cl, and then that is then put into the lumen of the stomach. And so together, those are what makes the stomach acid. Acid release creates the activation of pepsin, which is a digest proteins. It is also important for breaking down proteins before pepsin can digest the proteins. This just makes it easier and less surface area for pepsin, so it has to do less work. Again, this picture is from Silverthorne. Okay. As we continue, I will cover enzyme secretion. The stomach produces two enzymes, as we talked about in the last slide. Pepsin is one of them, and gastric lipase is another. First, I will cover pepsin. Pepsin carries out the initial digestion of proteins. It comes from initially pepsinogen, which is released by chief cells. As you can see down in this slide right here, this picture outlines exactly the path it takes to get to pepsin. The chief cells are stimulated by acid, as we talked about in the last slide. The gastric acid stimulates pepsin to be released, and then, which it is actually acting on the chief cells, which secrete pepsinogen, and then the acid comes in play to help with the pepsin. The gastric lipase is co-secreted with pepsin. Lipase are enzymes that break down triglycerides. So again, pepsin is the digestion of proteins, and gastric lipase is the digestion of triglycerides. Continuing, we'll go over the Pancreas Creation. First is histamine. Again, this picture from Silverthorne really goes over all of these. As you can see, histamine is present here and how it plays into it. It is secreted by the ELC cells, and they act on the parietal cells, which secretes acid. Next is the intrinsic factor, which is secreted by the parietal cells. And then the SS, which is a form of growth hormone, secreted by D cells, as you can see right here, and is a negative feedback from the acid. So that then acts on the ELC cells. So again, this picture outlines really well how all these interact. To review it, these inputs happen in the phase before the gastric phase. And then we have the G cells, which secrete gastrin, that act on the parietal cells to form the acid. The acid is important for the production of pepsin, and pepsin breaks down our digestion of proteins. And then you can also see here, as we move along, as we talked about the SS and the ELC cells on histamine, they all work very well and closely together to regulate digestion and regulate the secretions needed for digestion. Lastly, I will cover defense mechanisms and how it's regulated and why it's important for digestion. The stomach uses two forms of defense. One is the formation of mucus, or creating mucus via mucosal cells, and the second is the bicarbonate layer that is used as a chemical buffer underneath the mucosa. So mucus helps protect the mucosa layer, which is the outside layer in the lumen, the first layer that anything we ingest comes in contact with. Mucosa is just a barrier that protects it. And then the bicarbonate layer, when they say chemical buffer, it has to do with pH. The stomach has a very high pH. Bicarbonate, its purpose is to make things more alkaline. So that pH difference is a really good protective barrier for the stomach and regulating the digestive system. Why these defense mechanisms are important is it keeps our bodies at homeostasis, and at homeostasis we can digest properly without any issues. Alright, and then here's my study aid. It is matching terms. There's so many different ones to memorize, so this was supposed to be helpful with that. And then again, I got everything, all my information and images from Silverthorne.